Thank you so much for inviting me to speak today. I'm really excited to be here. And also want to thank you all in advance for sticking with me for the next 20 minutes, hopefully, and I can engage you with what I hope to be quite an engaging talk. So over the next 20 minutes, I'm going to talk to you a little bit about how Proteintech has evolved over the last 25 years from a traditionally polyclonal antibody company to a recombinant reagent provider, and how we're hopefully making waves in epitope mapping space to provide a new standard of antibody validation. So first off, I just want to take you back to 2001. So there's probably a few people in this room who have met our CEO and founder, Dr. Jason Lee. He had the idea in 2001 to essentially create a company to manufacture all antibodies in-house and validate all antibodies in-house with the main goal to cover the entirety of the human proteome and have an antibody for every target that works in every single application. And to date, we've covered about 2/3 of the human proteome. So there's about 21,000 human proteins, and we've got about 14,500 of those covered, and that is an ongoing process. So we've covered those with our polyclonal antibodies and our mouse monoclonals. And like I said, everything that we sell, we manufacture everything in-house, and we stick to the 5 pillars of antibody validation, which I'm sure there's a lot of people in this room who are instrumental in determining. So in 2018 was when we sort of pivoted a little bit, and we wanted to diversify our portfolio, and this is when we acquired Humankind, which is our branch of our portfolio that specialises in cytokines and growth factors. These are recombinantly produced, and it was the first time that we kind of entered into this recombinant space. They're produced in human cells, which means that they have native folding, post-translational modifications, which means they're really highly bioactive compared to those bacterially derived counterparts. It was at this stage as well that we got experience in GMP grade manufacturing. So we got our ISO accreditation, so we can manufacture our products from research use only all the way up to GMP grade, which meant that we could kind of enter a market in the translational space. So targeting those researchers that are lucky enough to be working in from bench side all the way to bedside. In 2020, the year that we want to forget, was when we acquired Proteintech. So Proteintech is our nanobody-based reagent portfolio. Nanobodies are why we have our alpaca on our table outside. These are variable domains of heavy chain only antibodies that are raised in camelid species, so camels, llamas, and alpacas. They're super tiny, so they're 10 times smaller than your traditional antibody, which means they're great for things like super resolution microscopy. They're really highly stable as well, so we've got products that are really niche for IP because they have really high affinity. Nanobodies can access cryptic epitopes that traditional antibodies might not be able to. So acquiring this arm of our portfolio really diversified us and meant that we could complement our recombinant offering. And that's what takes us to now. So thinking back 20 years, we were an antibody company at heart. We acquired some recombinant antibody manufacturing skills along the way, and this is where we come to with our antigen-specific B-cell cloning and engineering platform. I'm really happy that I managed to get that out. So the ABC platform is our recombinant antibody platform, where we produce recombinant antibodies with our wealth of experience from the last 20 years. So these are this is a really high throughput platform because we've got all of the PBMCs, all of the B cells from our polyclonal mouse monoclonal offering. They're super highly validated just because we do adhere to all of the 5 pillars of antibody validation. We've got a super secure supply because they're recombinant. You can send that sequence in an email. We don't need to go through any border control issues that quite a lot of geopolitical stuff might happen in the future. And also they're a reduced scientific cost because we essentially try and pass that saving on to the scientist. They're really super scalable as well because of the recombinant nature of them. So I'm really pleased to be able to talk to you about them today. So over the next 5 years, I've made this graph and I sent it to our CEO and I was like, is this right? And he was like, yeah, that's great. Essentially over the next 5 years, what we hope to do is cover the entirety of the human portfolio, the human proteome with our recombinant antibodies. We aim to have 3,000 targets a year covered, which forecasts us to, in the next 5 years, overtake our polyclonal offering and really do become the recombinant reagent company that you can work with. So that takes our goal from an antibody for every target, for every application, all the way to a reagent for every target, every workflow, and every application. So we're hoping to have an antibody that works for every protein, but also specialised for all of the different applications. So just to kind of hone in on the antibody part of my talk, All of our recombinant antibodies are sequence defined, which gives us real flexibility in the design of them. So your traditional IgG format is what everybody knows and loves, but it does come with some drawbacks. There's things like epitope accessibility, getting through cell membranes, half-life, etc., where other versions of an antibody might actually be a bit more beneficial. So because we've got the sequence definition of all of our antibodies, we're able to produce Fab fragments, Fab2 fragments, SCFVs, SCFVFCs, VHHFCs because of our nanobody portfolio, and also chimeric antibodies. So we're really hoping to broaden our portfolio of antibody binders with the recombinant engineering capabilities that we now have. And just to give an example of one of the products that is now commercially available and attributed to the fact that we can recombinantly engineer super easily, this is our FC0. So FC0 is essentially a backbone that we've got sequence defined, and we've been able to mutate the backbone at the Fc gamma receptor binding site so that it no longer binds to Fc gamma receptors. So if anybody works with flow cytometry, what you'll know is that you need to block your cells, stop your antibody binding to the Fc receptors non-specifically and causing a lot of background. With this mutation, that ablates that. With the recombinant engineering capabilities of this backbone as well, what we've engineered is an AccuBright site-specific conjugation, which essentially means that, can you see my mouse? Yes, you can. We can have complete control over the degree of labeling of our antibodies. So at this site, we can add fluorophores, enzymes, biotins, drug payloads, whatever you're interested in, and we know exactly where it's binding, how much it's binding, and we know that it's not binding at the site that's going to interact with your antigen. So this is just the only bit of data really that I've got in this talk, just to show that because we've mutated the Fc gamma receptor binding site, we completely ablate any flow cytometry background that you might be experiencing in your pipelines. This is also really useful for things like therapeutic monoclonal antibody development as well. So if you've got, if you're looking at producing a therapeutic monoclonal antibody, you've got Fc gamma receptors in your body, you don't want those antibodies to non-specifically bind and cause off-target effects. So what I've talked about there is fundamentally our portfolio of antibodies, and at our core is in-house manufacturing and validation. But what we wanted to do is essentially push the boundaries of antibody validation and add a new standard with our first-in-class 3D epitope viewing module. So you may or may not have seen this, but essentially what our wonderful scientists have done is essentially map the epitope that our antibody binds on the target and produce this open access tool for researchers to use. So researchers can find their favorite protein on our website, click on it, rotate, zoom, manipulate this 3D structure of their protein and find exactly where we've got antibodies that bind. Now this has been done for over 2,500 proteins so far, and we hope to map our entire portfolio. But for those that we haven't mapped so far, we do have an immunogen selector module where essentially you can find the domain that you're interested in, toggle that filter, and it will pull up the antibodies that are going to be useful for you. So generating epitope level insight is not an easy thing, and there's a lot of methods that you can use to do this. So I'm going to very briefly speed through this because I realise that I'm going quite over time already. But there's, yeah, like I said, multiple different approaches to identifying the epitope of interest. So firstly, we've got structural approaches, X-ray crystallography, co-crystallizing your antibody in your antigen, hoping that it crystallizes, hoping that it's not salt, and then trying to figure out the actual atomic level interactions between your antibody and your antigen. You've got NMR spectroscopy and cryo-EM. But all of these techniques are quite time intensive and can be cost intensive as well. So even though it gives you really high resolution data, there are other methods that you can use too. Biophysical approaches, so HDX-MS, so hydrogen deuterium exchange mass spectrometry, cross-linking mass spectrometry. These are labeling techniques using essentially labeling an APO protein, or APO antigen, and labeling antigen bound to antibody, quenching and seeing which portion of that antigen was protected by the antibody during the labeling steps. You've also got non-labeled approaches like SPR and BLI that can be used to generate epitope level insight. You've also got functional and fragment-based approaches, so peptide scanning, getting overlapping peptides with the entire sequence of your protein, using a ELISA assay potentially, where your antibody is binding or what hotspot of peptides that antibody is binding. You can then functionalize these peptides in a 3D domain and use yeast or bacterial surface display to look at it in its 3D conformation, and then use site-directed mutagenesis to validate that that particular peptide was actually responsible for binding to your antibody. And then good old-fashioned AI using to complement any of these approaches too. So AlphaFold is our Lord and Savior if you're a structural biologist, as I am. This is basically a technique, a machine learning model that has been used to solve the protein folding problem by just using the protein sequence. And this has been instrumental in helping Proteintech build the 3D epitope viewer alongside mining all of the structural data that's already been out there experimentally solved. So these are a few of the methods that you can use to generate epitope level insight, and Proteintech has used a combination of the above. We believe that having the epitope level insight is going to be fundamentally useful for making immunoassays that much better designed. And you know what, this is not going to play on loop, so I just want to make sure that people actually see this data set. There's many benefits that knowing the epitope can bring, so across all of the fundamental immunoassays. So knowing where an epitope is in its native setting, for example, if you're looking at a transmembrane protein, if you know that that epitope's on the extracellular portion of the cell, then in your IF pipelines, you're not going to need to permeabilize, and you might not have known that before. So this means that it opens up live cell imaging possibilities, for example. Even in IHC, so if you know that your epitope is riddled with lysines and arginines, that epitope is most likely going to be cross-linked during fixation steps. So if you know that that epitope has got a lot of lysines and arginines, you can maybe change your protocol, use a different fixative, or do an antigen retrieval step. You can also differentiate between isoforms. So if you know that the epitope of interest is completely homologous between two different isoforms or does have distinctions, then you can safely say that you can distinguish between them. You can use the knowledge to see if there's a post-translational modification within that epitope. You can also predict cross-reactivity. So if you're looking at a rare species protein, so I know a lot of our customers are using zebrafish, and not a lot of antibody companies will have proteins that have actually been validated with zebrafish. But if we've got an epitope that we've mapped on our human version of the protein, and you know that it's completely homologous with your zebrafish protein, then we can safely say that that antibody could be used for that. Comparability across assays and clones too, and also avoiding steric hindrance. So just on my next slide, I'm going to talk about matched antibody pairs again. So because we've got a lot of epitope level insight into our portfolio, what we've managed to do quite quickly is develop a portfolio of matched antibody pairs. So at the moment we have 3,600 human targets where we know that we've got at least 2 antibodies that bind to the same protein at the same time but don't sterically hinder. And as the talk previous to me was alluding to, this is instrumental for building lots of different assay platforms. We offer multiple pairing combinations as well, so you don't just have 2 antibodies, you could have antibody A binds with B or C or D. And this is great for ELISAs, for fundamental sandwich ELISA design, FRET-based assays, lateral flow tests, and others that I'm sure there's many people in the audience who are also building platforms. So if you are interested, please do get in touch. We're more than happy to help you out there. In the last few minutes, I also want to introduce our new tool. So hopefully what I've managed to do in the last 15 minutes is talk to you about how Proteintech has moved from predominantly a polyclonal company to a recombinant reagent provider, hopefully being really transparent with our epitope level insight. What we're also trying to do is push the boundaries of what tools we can give to researchers. And we are currently the first in the industry to provide an AI-powered search tool. So this was released a week ago. It's called Able AI. Reason being, our fantastic scientist is called Able, so we wanted to honour him. It's also AB, which is really useful. What you can do, it's a large language model. It's on our website. You can go on it. You can say, hey, I'm really struggling. My PI isn't telling me what to do. Can you help me design my experiment? I need to find an antibody for KRAS in this species. I'm using this buffer, and it'll pull information from all of the citations from our products. It'll pull information that we've taught it about a specific research area, and it'll give you a list of product recommendations and protocol recommendations as well. It builds on the epitope viewer information that we've managed to build over the last few years, and it also builds on the data that our wonderful researchers have been putting out into the scientific community. My favourite thing about this is also that it understands and responds in multiple languages. So what we're trying to do is make all of our products and our resources really accessible worldwide. And this tool really democratizes global research, we hope. So, final slide. So looking ahead, what we really hope is that our data transparency is really going to drive research progression. So going back what feels like a while ago now, we've got sequence-defined reagents, hopefully giving customers, researchers, the confidence that we can build and engineer different tools, so flexibility and custom options. We offer epitope and immunogen level insights, and also trying to push the boundaries in the kind of research tools that we offer in the form of AI. And I hope that's my last slide. Yes, it is. So yeah, thank you so much for your attention. I hope that was useful. And if you want to check out your protein, scan that QR code and more than happy to answer any questions.